As a student, I was taught that one should never begin a lecture with an apology. And so in following that, I'm going to start with two apologies. One, because my French is inadequate to give the lecture in the local language, so I'll have to do it in English. And the other, which is on this slide, which is that I'm going to talk about flower development and not about all of plant development. And the reason for that is that the time isn't adequate to talk about all aspects of plant development. And in any event, I work on flower development, so that's the part I know about. So with that gone, I will point out the basic problem of flower development, which is the same problem that one would try to solve looking at roots or embryos or even parts of non-flowering plants, is the question of how patterns form in development after the fertilization of the egg by a pollen cell, double fertilization that Mendel didn't know about, but was discovered in the 1890s. After fertilization, the egg begins to divide, and in a patterned way, its progeny become particular cell types, and it's a patterned and very dynamic way. In the flower, a simplification of this problem at the organ rather than the cell level is that there are four types of organs in most flowers: the sepals, the petals, the stamens, which have the pollen grains, and the carpels, which contain the ovules. So the simple question I want to approach is, how is it that a collection of plant cells knows enough to be able to make the correct organs in the correct place? That is the fundamental question of developmental biology, which is the question of pattern formation. I want to try to make 2 points while I'm discussing it. The first point is to give you a summary of some of the research in flower development that's gone on and an intimation of the sort of directions in which the field is going. But at the same time, I want to raise a historical question, which is why it took us so long to figure this out. Because as I'll point out to you, the experiments that Dujon just mentioned in this ABC model of flower development could easily have been done by Mendel. There isn't a single method that was used that he didn't have. Why didn't anybody do it? And I'll try and answer that question as well, at the risk of a long discussion with the actual professional philosophers and historians of science who probably have other ideas that I'd be very happy to hear about. Okay, so the problem of pattern and I really would be, because I'd like to know what the professional opinion on this is, and I'm only going to give my own personal opinion. This question of pattern formation and flower development is hardly new. Eichler in 1875, or one of my postdocs, and 100 years later, or more than 100 years later, we're asking how it is that in these mustard flowers, and those are the ones I'll talk about, Arabidopsis flowers, how it is that sepals surround the petals, which surround the stamens, which surround the carpels, which is a pattern that's universal in every known one of the 250,000 species of flowering plants, with two provisos. One is that all of those organs are present, because there are unisexual flowers that don't have all those organ types. And the second, there is one species, Lacandonia schismatica, a parasitic plant that grows in a very small region in the Lacandone jungle in Chiapas in Mexico, which does not have that pattern. And I won't have the time to talk about it, but I could refer you to the papers. All right, so what did we learn about flower development from Eichler or from Mendel to the present time? And between 1857 and 1990, I would say, probably controversially, that we didn't learn anything about flower development. And that's what the very last part of the talk will be about, why we didn't. There's Paillet, Jean-Baptiste Paillet, a member of this academy, and there's his description of mustard flower development in 1857. On the right, 1990, is a paper from my laboratory in which we didn't find a single thing that Paillet didn't already know. And indeed, our paper was information on a single species, while he had thousands of species in his book. So he knew much more than we knew. We'd actually lost information from 1857 to 1990. And the only thing you can say that was better about our work than what Paillet did is that we took photographs of the flowers in which you could see individual cells, even though you couldn't see anything about them, while Paillet did drawings, very artful drawings, of exactly the same phenomena and saw exactly the same things that we did. So what else could we do besides describe the development of flowers as Paillet had done? And we began to look at flower mutants, and I want to point out I'm showing a few mutants in which the flowers develop abnormalities, and they turn out to be the key to understanding the regulatory processes that create pattern in flowers. But these mutations were perfectly familiar in Mendel's day as well. Another member of this academy, Moch and Tandau, had in 1841 published a compendium of such mutations, although he didn't draw a distinction as to whether they were inherited or accidental. Of course, how could he have thought of that? But all these sorts of mutations were known by 1841. And in 1869, Maxwell Masters wrote a book which has a very modern point of view on these things, except again, without saying whether they're inherited or chance phenomena, in which he called Metamorphose, in which Bateson, who, as we learned, was a great master at coining terms. In fact, when I was at Cambridge, the best I could think of was I was professor of plant morphodynamics, which is not nearly as neat as professor of genetics, which Bateson wanted to be when he was at Cambridge. And of course, that title is now vacated since I've left Cambridge, while there is still a professor of genetics there. In any event, Bateson coined the term homeosis for these sorts of variants, which were called metamorphoses, based on Goethe's earlier work that I'll bring up in a minute, by Masters and by Bateson's predecessors. And so what we did was, starting with a wild-type flower collected mostly from colleagues, homeotic mutants of flowers that change the pattern of organs from the outside to the inside of the flower, in the hopes that by studying these mutations, we could understand the genetic basis for the formation of a pattern. Another philosophical question is how it is you can understand from things that are abnormal, things that are normal, a discussion I had for a long time with Don Kaplan, a distinguished plant morphologist, but that's a question for another day. I'll show you how. And so here's the wild-type flower, sepals, petals, stamens, and carpels from the outside to the inside. And among the mutations that we were able to find, both from our own mutageneses, and admittedly Mendel couldn't do mutageneses, but he had access to mutations just like these that had just arisen more slowly, and from colleagues who sent us flowers, was this type that we called an A-type mutation that has carpels in the first row rather than sepals, stamens in the second row, stamens in the third row, and then carpels again in the fourth row. So it's a mirror image of the inner parts of a normal flower, and that if we'd call the different regions of the flower, the whorls of the flower, A, B, C, and D, this represents the organ types that are found in D, C, C, and D, something like that. And this is a single gene mutation in a gene called APEDLA2. This is what we called the B function mutants, sepal, sepal, carpel, carpel. Instead of the 6 stamens, you have 6 carpels there surrounding the 2 central carpels, and they all fuse together, so you can't see the actual carpels in the middle of the flower. And we've peeled away one sepal so that you can see that the petals, the position where petals would normally occupy in the flower, are filled by sepals. And then the C function mutation has 2 things. It's indeterminate, so it keeps growing. When carpels aren't made, the flower keeps producing organs. The familiar double mutant phenotypes that have been known and described since the Tang dynasty in the 10th century in China. And it has sepal, petal, petal, sepal, and then that continues to repeat. So that's a mirror image of the outer whorls of the flower. What we did from this was proposed a model that we call the ABC model, and it's now been elaborated upon so that in various sources it's now the ABCD, the ABCE, the ABCDE, and so on. But this was the basic model, in which we proposed that the classes of genes represented by our mutations had a positive function that was revealed when their function was lost, and the positive function of A function genes is to specify sepals. When expressed in combination with B function genes, the combination A plus B causes the cells to become petal cells. And note that I'm talking about the organs as if they're unitary and not made of many different cell types. That's a problem we'll get to later. B plus C combinatorially specified stamens, and C alone specified carpels. And there's one more postulate of the model, which is that the A and C functions are mutually inhibitory, so that if one eliminates A function, C would spread through the entire flower, and if one eliminates C function, A would spread through the entire flower. So how does this fit with our mutant phenotypes? Well, if we knock out A function, C function spreads through the entire flower. B function is in the second and third whorls of the flower as normally, and so we get carpels, stamens, stamens, and carpels. Of course, I'm acting as if this is a prediction of the model, but it's hardly that because we already knew the phenotype when we made the model. I'll get to the predictions in a minute. But the model so far fits with what we saw in the loss of function mutations. A B mutant has only A and C functions, so it has sepal, sepal, carpel, and carpel, and nothing that can make petals or stamens. In a C mutant, because the absence of C, A extends through the whole flower, and the combinations of A and B create sepal, petal, petal, and sepal. And then that repeats because the flower becomes indeterminate, a second function of the C function genes in the middle of the flower. So the model fits the mutant phenotypes, but a proper hypothesis has to predict. And so one prediction is what would happen if we made an A-B double mutant by doing as Mendel did and crossing two mutant strains to each other. And the answer is that with B absent and A absent, C should be expressed everywhere in the developing flower. And consequently, we should get a flower that consists entirely of carpels, which is just what we do get. Or that if we make a BC double mutant, A should be expressed everywhere. There is no B or C, and so we should get a flower that consists entirely of sepals. And because there's no C function in the center of the flower should be indeterminate. And there it is, exactly as predicted. So the model has served two predictions that seem to be, the model's not been falsified, so we went on to run with it. And we did the obvious, we made the ABC triple mutant. Now I'll point out that the triple mutant, our model makes no prediction at all for what should happen there. But in fact, what we got when we knocked out all three of these functions is that all the floral organs are replaced by leaves, which again was something with which Mendel was perfectly familiar because Goethe in 1790 had predicted that all floral organs were derivatives of leaves and the organ was a leaf and that it was the progressive purification of the sap as it rose up the plant that invoked from the basic leaf plan these successively more perfect organs, the sepals, then the petals, then the stamens, and then the carpels. And so we didn't find anything that hadn't been known so far since 1790. And we didn't use any methods that weren't known in 1866 when Mendel published his paper. But we did get one thing out of this, which is that we were able to design on paper new types of flowers never before seen and then create them by Mendelian genetics. So he had done something and we were able to predict the outcomes of such experiments. We'll get back to why nobody else, somebody should have done this in the 1860s. Bauer had all the mutations in snapdragons in the 1930s. They could have done it. They never made a double mutant of two flower mutants. They always did one flower, one color. We'll get back to it. All right, so what do we now know up to this point in my talk about flower development? Everything I've described could have been done by Mendel. There were no methods used that he did not have. Of course, this didn't last for very long. We've heard about the sequencing of the Arabidopsis genome done in part in Genoscope. And so needless to say, we availed ourselves of the knowledge that genes are DNA and of the armamentarium of modern molecular biology that we heard about in the first lecture and cloned some of these genes. But even without cloning them, we have the ability to design flowers with new patterns of organs and then to grow them due to a tested mechanistic hypothesis, the very abstract hypothesis that involves regulatory genes that interact with each other and then create organ types by acting somehow within the developing flower primordium. So we cloned some of these genes, and what we found out when we cloned the genes is that all of the ones I've shown you and many more that I haven't shown you all code for DNA binding proteins. And so the prediction is that all of them bind to DNA and regulate other genes, which but before I get onto that, let me point out that the fact that we cloned these genes, or members of all these classes of genes, enabled us to create some additional tests of this ABC model of flower development by gain of function rather than by loss of function. And so that, for example, if we express a C function gene, not only from its wild-type copy, which is in the genome, but also when attached to a B function promoter and transformed back into the Arabidopsis plant, we would predict this pattern of gene expression where we have A function in the first world, but because we've artificially overexpressed C in the second world, that should shut down A. And then the combinations of A, B, and C should create a flower that has sepals, stamens where the petals ordinarily would be, stamens again, and then carpels. And I wouldn't be saying this if we hadn't done it and couldn't show you that that's exactly what happens. And so we can replace each petal neatly with a single stamen. Or if we express B everywhere, there are two B function genes. I didn't show them both to you, but if we take both of them and express them, the pistillata and apetalate3 they're called, and if we express them both from a promoter that's expressed everywhere in the developing flower, here's the prediction of the ABC model, which is that we should get petals replacing the sepals, exactly what you see in tulips, where the B function genes are expressed in the first world. Petals, petals, then stamens and stamens, and here are these lovely things, exactly according to the prediction of the model. Or what if we have A and B mutants everywhere? So we have now a triple trend. Well, we have an A function mutant and our two B function genes expressed everywhere in the developing flower, and we should get a flower that consists entirely of stamens. And this isn't such a flower. What this is the entire inflorescence, picture taken from the top. And each of those groups of stamens is an individual flower. All that we ever get once the plant starts to flower is this manly thing, a flower made only of stamens. Many such flowers. And so the model holds up quite well under loss of function and gain of function mutations, and it gives us the capability of creating new types of flowers. It doesn't answer very many questions about the mechanism of pattern formation and development, but it does answer one question, which is that by the spatially appropriate expression of certain regulatory genes, the patterns result. Then the question becomes, what causes these regulatory genes to be expressed in the appropriate spatial patterns? And then secondly, what do these regulatory genes do to allow the cells in these developing organs to turn into the cell types that are characteristic of the different floral organs, rather than the cell types that they might if we hadn't interfered with things. And so many experiments have been done. This is just one showing that the proteins encoded by these genes all bind to DNA, and they bind upstream of particular genes and activate or inactivate, repress those genes, and so they create a new pattern of gene expression in those cells where they're expressed. And this is up to about 2010 and is just a piece of a figure from a paper published in the lab of one of my postdocs, Frank Welmer, and Jose Luis Reichmann, another postdoc, and also my lab participated, in which we attempted to map out the gene regulatory network that activates these genes, and then in other parts of the figures that is activated by these genes. And this is one narrow piece in the temporal path from flower primordium to mature flower, showing positive and negative feedbacks both between steps at different times and between steps that are activated at the same time. And so we come to a very complicated picture of gene interactions, which is the real picture of what's happening. And as I'll point out in a minute, to be able to deal with these complicated pictures, we have to develop computational methods, because we can't any longer appreciate a figure like that in our heads and understand what these feedbacks, both positive and negative, and also feed forwards, both of coherent and incoherent types. Many different sorts of network motifs are exhibited by these gene regulatory networks. And to really understand flower development, we're going to have to understand at a fundamental level not only what the networks are, but what the consequences of the feedbacks and feed-forwards in the networks are. Here's another more attractive way of putting it in a review article written by a former postdoc of mine and a postdoc of his, in which they point out that the earlier expression of genes like UFO or Wuschel and leafy in the earliest primordium of a flower lead to the later expression of the ABC genes, which I discussed, and then the activation, those genes act to activate further downstream genes, and they do it by making multimers, tetramers, which is how they act combinatorially, is that a tetramer of apetalate3, pistillata, agamous, and sepillata3 does something different from one of agamous, and sepillata3, so that these tetramers activate or repress different downstream genes, and they lead to the specification of organ pattern. Now, what happens after these genes are expressed in their appropriate domains? Well, there are subdomains that are specified by further regulatory genes and sub-subdomains in which finally the genes in the final differentiated tissues that give them their function are expressed. And here on the left is an example of a gene that is activated downstream of apetalin-3 and pistillata, and that's expressed only in the tapetal cells, one layer of cells in a developing stamen, in the anther. Or another one on the right, which is expressed only in the filament of an anther at a very early stage of development, very transiently. And I should say that there was no recognition before this that there was anything different about those cells and the cells around them. They look the same, but they have a different pattern of gene expression, and it leads them to do different things later. So here's an example of one of the downstream genes just to show that often they're also regulatory genes. Sporocytlus nozzle is one of these genes, and it turns out when you knock out sporocytlus, there are no sporocytes, the anthers develop, but there are no pollen grains formed in them. If we overexpress sporocytlus, for example in a petal, as shown in F and G there, we can cause pollen grains to form in a developing petal. It's still a petal, so we've not changed the pattern of the entire organ, but we've taken a subdomain of that organ and made it develop as it would if it were a stamen. And the reason why a stamen does that is because this gene is activated in a stamen and isn't ordinarily activated in a developing petal. And so that we can say agamous, our C function gene, activates sporocytlus nozzle, and then that activates another set of genes which leads to microsporogenesis. So bit by bit we can peel back one layer after the other layer in flower development that includes a lot of communication within each cell of regulatory genes regulating each other and a set of genes that come on later. Those genes regulate each other and sets of genes that come on yet later. And in fact, because the life of the plant is a cycle, later eventually comes back to the embryo developing in the ovule and starts the whole thing over again. And it's always later, and it goes on infinitely till extinction of the species. Now, I want to introduce another aspect that I haven't talked about yet, which is the interaction of these cells with each other. I've talked about these patterns as if they come on in some domain of a continuum. I didn't say anything about cells. And then they create new patterns as the genes regulate each other in cascades with lots of feedbacks. But in fact, the cells are dividing all the time these genes are being expressed. And when they're being expressed in different places in developing flowers, this is a shoot meristem, the stem cells that create the flowers, and the little bumps on the side are the flowers as they first begin to develop. And so we had to develop methods to look at the dynamics of these developmental patterns by following the cell divisions and then labeling within the cells the expression of each of these genes so that we could see exactly the timing of them and then be able to intervene in the development of the plant to prevent or create new types or old types of cell-cell communication. And I'm not going to have time to say very much about that, except to say that there are two types of cell-cell communication that are critical here. One is chemical communication, which in plants is mostly mediated by peptides, which bind to transmembrane receptor kinases that activate downstream processes. There are plant hormones. There are 5 classical plant hormones. There are 5 new plant hormones that are small molecules, and there are 1,000 peptides. That's how plants communicate. But there are also small molecules, auxin and cytokinins, and the classical hormones we all learned about in botany communicate between cells. There's a second modality of signaling that I won't have time to say anything about, but turns out to be just as important as chemical signaling, and that's mechanical signaling between the cells. When a plant cell, which is surrounded by a cell wall and fused to its neighbors in such a way that the cells cannot slide with respect to each other, when a plant cell starts to expand, it puts its neighbors under stress, and the stress on the wall is interpreted by the neighboring cells to change patterns of subcellular protein expression and to change patterns of cytoskeletal organization, which in turn changes patterns of cell wall synthesis and cell division, which then affects the stress pattern, which feeds back again into the behavior of the cells. And that's just as important as chemical signaling. So let me show just a couple of things. So here's an example now of the sort of live imaging that we can do. And this is just one frame from a movie showing the expression of some of the genes I talked about before in radial patterns in a developing flower. And while our ABC model predicted that hypothetically, we can see that it's quite real and that the genes do express exactly as predicted. There's another one that I put up just because it's rather attractive. Some other genes. We've also developed computational methods to take these movies, like the one I showed you, and to follow each cell over time so that we can get a complete clonal analysis of the origin of each part of the plant and run the movies backwards to see where each organ originates in a single cell or in a group of cells. And we can do this now in an automated fashion. We've done it in the shoot apical meristem, and here's an example where we've done it in early flower development so that you can see the clonal growth of individual cells. And so that enables us to know what the substrate is in which these genes are acting. And to give an example then of the use of computational methods, which we now need because, as I used to say, not even jokingly, that we stopped at A, B, and C because we couldn't keep in our minds any more than the activity of 3 genes at once. But now we know about hundreds of genes, and so we need computers, which are smarter than we are, to keep track of everything that's going on, which we can't do in our heads. And so we approached another problem in the pattern of flower development, which is how this set of stem cells at the end of the stem creates flowers in the spiral pattern in which they're seen in a Arabidopsis plant, with each primordium coming out about 120 or 130 or 140 degrees radially around from the next one, just as in a sunflower or a pine cone or anything else. And you all remember this has something to do with Fibonacci patterns, which I won't say anything about. It does and it doesn't. In any event, we wanted to understand this spirophylotactic pattern that then leads to the patterns of organs in developing flowers. And this is an old problem of mathematical biology that also is perfectly familiar to Mendel, or should have been, because at the same time he was publishing his work, Hofmeister, who is the 3rd greatest botanist of the 19th century, Darwin, Mendel, there's the 3rd guy, Hofmeister. I would even start about what he discovered. It was a lot, and including that he made the first mathematical model of phylotactic pattern. Alan Turing, at the time he killed himself, was working on phylotactic pattern and invented the concept of a morphogen and reaction diffusion mechanisms to try and understand phylotactic pattern. Turns out that's not how it works, but those functions act in other parts of development, just not this one. And even members of this academy, like Yves Coudert, is the most famous and successful modern exponent of phyllotaxis in the mathematics of phylotactic pattern, with whom we've had many productive discussions. And so what it turns out is the heart of phylotactic pattern is this, that the hormone auxin comes to be expressed at high levels at each point where 2 days later a flower will form, and that's causal in the initiation of a new flower. Auxin is indole 3-acetic acid, which Darwin and Sachs wrote about in 1880. Again, nothing new, and it was shown to be a chemical substance of more or less low molecular weight in 1926 by Wendt. But it accumulates to high level in these particular regions, and the reason it does that is that it's transported actively across the epidermis of the tissue, and the active transport is mediated by a set of proteins that are asymmetrically disposed around the plasma membrane of the cell. So they'll pump auxin out of a cell in a particular direction, and that creates a circulatory system for auxin that's its own system that puts the auxin at high concentration in the phylotactic pattern. Here's a movie of that auxin transporter, and while with this lighting and at this magnification you can't see it, we can watch this movie. And first of all, the gene is auxin induced, so everywhere where the auxin concentration is high, the picture is brighter. If we focused in a little bit more, you could see exactly the distribution of this protein around each cell, and from that we can infer the direction of auxin transport at any time during the development of the shoot apical meristem. Based on that, we can create models which describe that, and this is one of the models in which we have the auxin concentration in each cell being a function of its synthesis, its degradation, the reception by that cell of auxin from its neighbors, and the delivery by that cell of auxin to its neighbors, the concentration of auxin in the cell wall between 2 cells, I and J, which depends on the action of these auxin efflux carriers, of additional auxin influx carriers and a diffusion in the wall, the concentration of the auxin efflux carrier itself, which is auxin dependent, and the direction on which the auxin efflux carrier is pumping the auxin from each cell. And if we take these and put them in our computer in a simplified model of dividing cells in the meristem, we get exactly the spiral phyllotaxic pattern and exactly the concentration changes in auxin that we see in our movies. And so now we're at a different level of understanding, and this is the model before we added mechanical force. We had a proxy for mechanical force in this particular model, but now we've added mechanical force to that, and we can, in great detail, represent exactly what we see in our movies of developing flowers, and now therefore understand at some level the phyllotaxic pattern, though there's more, of course, than what we've shown here, because the pattern is a little bit more robust than what we get in our computer models. So there's some feedbacks there that we're still hunting. So it gives an example of where the field has gone today after Mendel and applications of Mendelian principles. We already had control of flower development and a certain understanding of how pattern in the flowers is created at the organ level, but we had to take the microscopy to a level that was not known in Mendel's time with reporter genes, transgenic plants, DNA sequencing, and computational modeling to get to the next step, which is how the cell-cell interactions create pattern at a finer level than the whole organ. And that's where the field stands now and is moving forward very rapidly in a great many labs. I should point out what I should have said at the very beginning. Why is it even important that we do this? And I'll point out simply two things: that flower development and plant development creates our food, and the population of the earth is going to be 25% higher in about 20 years, and the amount of arable land goes down about 1% a year as a result of urbanization and desertification. So we have less land in which we have to grow more food. Another reason why it's important to study this is that plants are the reservoir of carbon outside the atmosphere. There's almost as much carbon stored in plants on land as there is in the atmosphere. And so that if you could increase carbon uptake or decrease carbon output from decaying plant material just a little bit, you can influence the entire atmosphere. All right, so it's important enough. And now the models have finally gotten to a point where we're modeling not just the epidermis, but the whole three-dimensional shoot apical meristem. And this includes some components I didn't discuss at all, but a direction we're pushing this is to be able to completely represent the gene regulatory networks that create the different cell types in the shoot apical meristem, that create the floral primordia that surround the shoot apical meristem, and then that create the pattern collections of cell types in the developing flowers that are equivalent to the floral organ types. And one step at a time. I don't think we'll get there anytime soon, but I think we can get there one step at a time. Now, let me get back to the historical point that I promised you at the very beginning. Why is it that Mendel didn't do this? He could have. Bauer could have and should have. I can't believe Bauer didn't do it in his lab. There's a guy in his lab named Brieger who even wrote a little paper in which he said that they could do it, but they didn't. He was... well, let's say, I won't start a disquisition on European history, but he was compelled to move to Brazil at a certain point in the history of Germany and became an orchid taxonomist. We only have 3 theories in biology, as far as I know. If there's another, somebody can tell me. There's the theory of the cell that Nut Schleiden proposed in 1838, the theory of evolution, on which I haven't even touched, but we all know about Evo Devo and where I could go with that if I were to speak about it. And then Mendel, who created the theory of the gene, which was elaborated under that name by Morgan, but in fact that's what he began doing, was creating the theory of the gene. And developmental biology requires consideration together of postulates that arise from the theory of the cell and the theory of the gene. And then in its full manifestation, also the evolution of development, it requires all 3 theories. So one had to not only elaborate each of these theories, but be able to think about more than one of them at a time to get anywhere with it. So in most organisms, development is a cellular phenomenon which cells divide and differentiate in patterns. There's some organisms where that's not true, and I want to talk to the algologist here about some of these later. Caulerpa, just one cell. Boy, it's just as differentiated as these flowers. The formation of these patterns and cell division control could be studied by inferring mechanism from genetic mutations, though, was not obvious to Mendel or to Bauer or to other people who worked on flower development, like Stebbins in the 1960s. It was not obvious that you could infer mechanism from genetic mutations. And so the application of Mendelian methods and concepts to development began in Drosophila and C. elegans in the 1970s and got to plants very rapidly by the 1980s. Why is that? Why was the connection of genes to development non-obvious? And I would propose that this is because the conceptualization of the gene that we heard about in Guéant's lecture began with the work of Beadle and Tatum, which showed that genes coded for enzymes. And enzymes are catalysts. And so that genetic effects in evolution and in develop were therefore attributed, for example, in Stebbins' work, to changing enzyme activities in cells. And the pleiotropy of developmental mutations was explained as a necessity for broadly used metabolic processes. So if you affect the synthesis of a vitamin or amino acid, you could have pleiotropic effects in development, but that wasn't actually what was happening. It wasn't the enzymes, it was the concept of regulatory genes. And it was only with the work of other members of this academy, Jacob, Monod, and their colleagues, that it became clear that genes were not only coding for enzymes, they were coding for regulatory factors. They thought some of theirs were RNAs, they were proteins, but they thought some could be RNAs, and they later turned out some are RNAs. And so with the concept of regulatory genes, the rest followed. And McClintock emphasized regulatory genes in plant development and in development of seeds, but her conceptualization of it wasn't quite the modern one. With the Jacob-Monod concept of regulatory genes, the rest followed. Transcription factors as informational mediators of gene expression, including feedbacks between regulatory genes, all arose from the work on the lac operon and other bacterial operons, and was only when that conceptual framework was applied to the development of multicellular organisms that any progress could be made beyond what Mendel had done in looking at his flower mutants. And so that I would say that it wasn't a development of a technique of any sort, or of a new method, or of anything of that kind that enabled us to begin understanding development at a genetic level. It was a conceptual advance, but of course to maintain that conceptual advance required DNA sequencing and confocal microscopes and every other $100,000 piece of equipment that our sad laboratories have to have now. So I would say that as a conclusion, the genetic approaches to development and to flower development only bore fruit after a conceptual advance, and not initially, at least, due to a technical advance. And I would leave that open now for discussion by the professionals. And I just want to show, it's hard to read, it's a list of names of the collaborators and people in my laboratory at the moment, and some pictures of some of the people in the labs. The lab I had when I was at Cambridge on the bottom, and the one I have now at Caltech at the top. And of course, because we need those $100,000 or now $500,000 pieces of equipment, most of all thanks to the list of names at the bottom for the people who provide the money for my laboratory so that we can be part of this modern adventure in developmental biology. So I leave the floor open for questions, and I warn you, if they're in French, I'm going to ask Anne to translate them for me.
Well, thank you very much. Question, puisqu'il y a des en français ou en anglais, les deux sont possibles. Oui.
Are the genes, the ABC developmental genes, specific to plants, or are they akin to developmental genes in vertebrates, or are they totally different genes?
No, they're not. Most of the ones I showed are members of the Madsbach transcription factor family, which is also found in animals. Serum response factor, for example, is a Madsbach genes, and they're ones involved in heart development in vertebrates. Now, when I say they're members of the same family, they have the same DNA binding motif, clearly evolutionarily homologous, and they bind to not only similar, but identical sequences. We've replaced the binding sequences in our genes with the ones from human serum response factor, and they still work fine in the plants. The rest of the protein, though, is kind of different. And so I'm shading the truth when I say that they are the same as vertebrate genes. The other one I showed, the A function gene, APEDLA2, that's from a transcription factor family that is not known in animals or in fungi. You get both kinds. About half the transcription factor families in plants have a DNA binding motif that's clearly present in animals or in fungi, and the other half are plant specific.
Autre intervention, quelque part. Où est-ce qu'il est? Ah, là-bas. Oui, Michel.
Thank you very much for this nice presentation. You showed that the ABC model works in dicots. And my question is, does it work also in monocots?
Yes, there's some variations. There are two C function genes, so you have to get both of them. And of course, in grasses, the petals actually develop as loticules that are rather different, and the glooms are like the sepals, and so there are additional factors in that. But fundamentally, yes, it does. Lacandonia, in fact, is a monocot, the one that has the organs in the wrong place. And that can be shown to be due to evolution of the expression domains of some of these ABC genes. So yeah, pretty much the same. There are differences. I mean, you know, there are more or less genes doing the same thing. There's subfunctionalization, neofunctionalization. Petunia, for example, another dicot, has 3 B function genes, one of which is petal specific. And so it's not exactly the same, but the basic idea is the same in all the flowers we know of.
Une question ici. Here at the back. At your back.
I have
Your
a
back.
question right here.
Your back.
My question is about your modeling that you showed. Where are the genes entering in this model? And what is the spatial pattern that how do you model the spatial patterns that you've shown in the calculations here?
Okay, the PHYLLOTAXIS model, the only gene product that's in there is the auxin efflux carrier. And we abstract all the many genes that are activated and repressed by auxin itself, just by making the cells turn yellow and saying that's a flower. And so, how do we model it? Each cell is running all those differential equations over and over again. And indeed, you can start with them all uniform in their auxin concentration, and rounding errors eventually create the phyllotaxic pattern, so that the pattern arises naturally from any asymmetry in auxin concentration. And the cells are dividing, and they're communicating with each other only by sending auxin into the wall, which then goes into the next cell. Now, the next model I showed with the 3-dimensional meristems that I didn't describe also includes diffusion of the hormone cytokinin from the top level farther down into the plant. It includes the appropriate expression domain of the cytokinin receptors and of downstream genes activated by the cytokinin that activate further cytokinin-regulated genes, including regulating the receptors, and all that is built in. But again, it's each cell on a separate computer model that represents the 3-dimensional structure of the meristem, that all those cells are connected to each other in that model by chemical communication. That still doesn't have the mechanical part. That's another set of models. And the division is a pretense to be like the division. They're just bubbles popping, to be at the rates it is in a real meristem, but the division patterns aren't part of the gene regulatory network yet in those models. So basically, each thing's a bubble with a bunch of differential equations in it, more than what's seen there. Some asymmetry, either that occurs by rounding or because we start with initial conditions. The epidermis is the place where the cytokinin is made. And then we let the model run. And if it doesn't do what our plants do, we work on the model a little bit. And then we change, and we're careful to write the equations such that the parameters are things we can change in our experiments. We could have written a much more simple set of equations for phyllotaxis than that. Couder has done, but not one that we could have tested in our plants.
Oui. Encore quelque chose? Non? Ah oui.
Standing
Okay.
between everybody
Ici.
and drinks.
There is one
Yeah, it's
question
a
here.
perilous place to be.
One
There's one
question
back
here.
here too.
And
How do you know that vegetal cell is a stem cell, since most of them are totipotent? Do you simply mean that they are stems in undifferentiated cells in a meristem, or more than that?
More than that. In the meristem, I mean, it's a set of stem cells in the sense that at the very top of the meristem, the cells can have any fate if they develop normally in the plant, if you leave them alone. And then their descendants, so they're pluripotent stem cells, their descendants are multipotent, and then the fate to which the cells will go continues to be subdivided from that initial population, which divides out and creates the top of the meristem. Then the cells are progressively specified into flowers, and then into floral organs, and then into cell types in the floral organs. And so there's stem cells in that sense. It is true that you can carve a plant up and get any cell to create a new shoot apical meristem, because the shoot apical meristems are not any cell. I'll take that back. There are cells that will make shoot apical meristems. It's the literature of plant biology says that any cell will do that, but it's actually not true. There's a special collection of adult stem cells that sits paravascular that are the things that make the new meristems in a plant, the xylem pole pericycle cells. And there's some epidermal cells that'll do it. The reason I'm hesitating is that is true, what I've said. If you cut the plant up in the traditional way with a razor blade, there's only one cell type, really, that'll grow out of that. And so they're not pluripotent, although no one has shown that if you make protoplasts and take the walls off the cells, that those are not pluripotent, which is also what the old literature says. And so probably all plant cells are not stem cells, maybe unless you remove the cell wall. But I call these stem cells just because that's their function. It's a set of self-reproducing cells that has many fates, and their progeny have fewer fates, and their progeny have even fewer fates. And so they act as pluripotent and multipotent, and then much more specific stem cells.
Ok. Oui, ici, oui.
I have a more general question. For the future of plant biology and plant development, do you think that we need more of a technical advance or conceptual advance?
Well, you can see the side of that argument where I always find myself. I think we need more conceptual advance. I think there are principles there that we don't yet recognize, and that when we do recognize them, the progress will come very rapidly. Now, I wouldn't claim that we don't also need technical advance. If I didn't use technical advances, they wouldn't fund my grants anymore, I guarantee you. And so at a minimum, I have to pretend to be using these expensive microscopes. In fact, we really do use them, and the level of information we have from these sort of technical advances is probably critical for conceptualizing the conceptual advances. I think it's a cycle, a virtuous cycle of conceptual advance that demands technical advance that then allows new conceptual advance. I have my favorite part of that cycle, but that's just me.
Ok, merci beaucoup. Je crois que ceci clôt notre cérémonie d'ouverture.